Hello everyone, my name is Mohammed Fazel and I'm a postdoc at ASU in Pressay Lab. In this presentation I'm going to talk about our recently published paper in ACS Photonics with title High Resolution Fluorescence Lifetime Maps from Minimal Photon Counts in collaboration with the lab of Professor Gratton and Professor Dick. In fluorescent microscopy, the target structures within cell are stained with fluorophores. In this diagram, you see a simplified quantum structure of such fluorophores. Fluorophores are often in ground state, but they can absorb a photon from illuminating laser, go to the excited state, spend time tau in the excited state, and then emit a photon and go back to the ground state. The time that that different fluorophore species spend in the excited state are different, which can be used to distinguish different structures within the cell. So here on the screen you see a sample that are stained with two fluorophore species, the blue one and the red one. People often use a scanning confocal microscope to acquire data from such sample. Each spot within the sample are illuminated with a trace of laser pulses in which a few of them can lead to photon detection that are shown here by the curly arrows. The acquired photons can then be organized into a pixelated image like what you see here on the screen. This is the raw film data, and there are different film data analysis techniques that can be used to distinguish the two different structures and learn their underlying lifetime maps. Such film experiment can be used to map variation in different parameters within the cell. For example, variation in temperature, variation in refractive index, variation in pressure, and variation in the concentration of different biomolecules. Inspecting the variation maps of these different parameters within the cell is important because they can indicate cell dysfunction due to different disease, for example, cancer. However, there are different problems, there are different challenges in film data analysis. The first problem is learning the number of lifetime components present in the film data. The second problem is dealing with a wide range of lifetime, for example, lifetimes larger than the interpolate times or lifetime shorter than the IRF, and also distinguishing lifetime with sub-nanosecond differences. The other problem is providing lifetime maps with subpixel resolution. The current film data analysis techniques are limited to pixel size. The other issue is the data inefficiency. Some of the existing methods bin the data that cause losing a lot of information, and also they don't use information from empty pulses. And the last issue is reporting uncertainties over all the learned parameters. In a previous work, we addressed the issue 1, learning the number of lifetime components for a single pixel. In this paper, we are going to address issues 2 to 5. In this slide and the next 2 slides, I'm going to present the mathematical framework that we propose to solve issues 2 to 5 that I explained in the previous slide. So first, I start from the data or observation from such film experiments. So the first observation is if a pulse leads to a photon detection or not, or in other words, if a pulse is empty or not, which I show here by W. The second observation is the photon arrival times, which I show here by delta T. They're going to be, my likelihood is going to be the probability of observing this data given the set of unknown parameters, theta, and the total likelihood is simply the product of the likelihood for each of these observations. So the set of unknowns that I show by theta are the lifetimes and the associated lifetime maps, and there are a few intermediate parameters that I'm not going to talk in details here. So the main object that I'm going to work with later is the posterior, which is proportional to the product of the total likelihood and the priors on all the unknown parameters. So the first unknown parameters is the lifetime maps, which is a set of infinite random variables that are correlated. Therefore, I use a Gaussian process prior on that. And I'm gonna use appropriate conjugate priors for the rest of the parameters. In this slide, I'm gonna talk about the likelihood of the photon arrival times. So on the top, you see the trace of illumination pulses used to excite the fluorophores. So for the sake of explanation, I'm gonna show one of these pulses by a box centered at TK. So let's assume that during this pulse, a fluorophore gets excited at time t excitation. And then it emits a photon after n pulses. Here the capital T shows the interpulse time. And then the emitted photon is detected by the detector by a delay. And the detection time here is shown by T detection. The observation is shown by delta T here, which is measured with respect to the immediate previous pulse. The time that the fluorophore spent in the excited state is shown here by delta excitation, which is the period from when the photon gets excited until the fluorophore emit a photon and go back to the ground state. There are also two other times. The difference of the center of the pulse, of the excitation pulse, and the time that the fluorophore gets excited, we show that by delta T IRF, and then the delay in the detection of photon, which I show by delta IRF2. So the sum of these two parameters is shown by delta IRF, or which stands for instrument response function. So from the above figure, I can write this equation, and then if I take nt to the other side, I arrive at this equation, which says that my observation delta t is sum of three random variables: the time that the fluorophore spent in the excited state, the time the IRF time, and the number of the pulses. So therefore, the likelihood of delta t is going to be the convolution of the likelihood for these three observations. In this slide, I'm going to talk about the likelihood of photon detection, which I show by W. To do so, I start by the fact that the number of detected photons from a pulse has a Poisson distribution. So if we assume the pulse is empty and there is no detected photons, then I arrive at this exponential expression here, which gives the probability of detecting no photons from fluorophore species of type M, where PSF gives the PSF of the confocal setup, and mu rho stands for the lifetime maps. So the probability of detecting no photons from all the species is simply given by the product of this P naught N for all the species present in the data. And then now that I derived the probability of detecting no photons from a pulse P naught, I can proceed to write the likelihood for W. W is a binary random variables reminiscent of flipping a coin. Therefore, the likelihood is a Bernoulli distribution, where with the success probability of probability of detecting photons 1 minus pi naught, where pi naught was probability of detecting no photons. Okay, so given all these likelihoods and the priors, my posterior is going to attain a non-analytic form, and I cannot analytically solve this posterior. Therefore, I'm gonna use MCMC techniques to draw samples from this posterior. So in this slide, I'm gonna talk about the MCMC technique. So let's say the posterior distribution that I have has this shape has the shape that is shown on the screen. I start from a random location, θ1, with the posterior probability, where the posterior probability value at this point is P of θ1, and then I propose moving, and then I propose moving to another point θ2 with the posterior value P of θ2. So I would accept this proposal with a probability given by the ratio of these two posteriors. Let's say I accept this proposal and I go to the second point, θ2. Then after that, I propose going to a third point, θ3, and then so on. I go to θ4, θ5, and I just keep doing that. So if I take enough samples, at the end, the distribution of these drawn numbers are going to be the same as the distribution of the posterior distribution that I have, which I can use for subsequent numerical analysis. So now I'm going to use the MCMC technique that I just described to learn the lifetime maps of the data that is shown here on the left using the Gaussian process prior. So here you see I start from a random lifetime maps, and then the MCMC chain iteratively converges to the true underlying map. So I took 40,000 samples here until the chain converges to the true map. I'm just going to wait until the animation ends. We used our proposed framework to analyze simulated and experimental FLIM data. Here on the left, you see simulated ground truth lifetime maps, and on the right, you can see what our algorithm learned, and they match pretty well. So we simulated data using this ground truth with different lifetimes. In this slide, you see lifetimes that are only separated by 0.5 nanosecond, and our algorithm can still learn this lifetime and distinguish them. Here the dashed line shows the ground truth, and the histogram shows the sample lifetime from the posterior by our algorithm. In this slide, you see lifetime shorter than the IRF, and our algorithm is still able to recognize and learn that lifetime. In this slide, we simulated data with a lifetime larger than the interpulse time. So interpulse time here is 12.8 nanosecond, and we simulated data with a lifetime of 25 nanosecond, and our algorithm still learns that. We also tested our algorithm using experimental data from lysosomes shown in green here and mitochondria shown in red. We acquired data from the mixture of these two lifetime maps shown in yellow, and then we use our algorithm to process this data and then recover the underlying maps, which match with the ground truth with less than 5% error. Okay, thank you for your attention, and I hope that you enjoyed this video, and our paper is published on ACS Photonics, and also it's also can be accessed on Fazel lab at ASU webpage. Thank you for your attention.